So, welcome to the class of recombinant DNA technology. In this chapter, we will be going through some of the basic techniques and processes that are involved in recombinant DNA technology. And at the end of the chapter, you will be able to learn isolation, quantification, and analysis of nucleic acids, the basics of molecular cloning, different techniques involving recombinant DNA technology, transgenic animals, downstream processing like isolation and purification of proteins, and the basics of cell culture techniques. All living organisms, from single-celled bacteria or protozoa to multicellular plants or animals, must store, replicate, and transmit to the next generation, and use these vast quantities of information to develop, reproduce, and survive in their environments. We know that DNA is the fundamental information molecule of life, which is transcribed to RNA, which is a mediator that converts or translated the genetic information to a functional molecule of life process, that is protein. The whole process is unidirectional, except for retroviruses, which actually make the DNA from RNA to infect the host cells. We will now quickly refresh the basics of DNA structure. For nearly 4 billion years, the double-stranded DNA molecule has served as the bearer of genetic information. DNA can withstand a wide range of temperature, pressure, and humidity and remain relatively intact for hundreds, thousands, or even tens of thousands of years. DNA is a long polymer composed of subunits known as nucleotides. Each nucleotide consists of a deoxyribose sugar, a phosphate group, and one of the 4 nitrogenous bases of purine or pyrimidine. Phosphodiester bonds always form a covalent link between the 3' carbon of one nucleotide and the 5' carbon of the following nucleotide. The consistent orientation of the nucleotide building blocks give a chain overall direction such that the 2 ends of a single chain are chemically distinct. At the 5' end, the sugar of the terminal nucleotide has a free 5' carbon atom. Free in that sense that it is not linked to another nucleotide. At the other 3' end chain, it is the 3' carbon of the functional final nucleotide is free. X-ray images showed that the molecule is spiral shaped. The spacing between repeating units along the axis of the helix is 3.4 angstrom. The helix undergoes one complete turn every 34 angstrom, and the diameter of the molecule is 20 angstrom. Terms such as molecular biology, genetic engineering, and recombinant DNA technology are sometimes used interchangeably and often mean slightly different things to different people. Molecular biology, in its broadest sense, describes the study of biology at a molecular level, but focuses in particular upon the structure, function, and interaction or relationship between DNA, RNA, and proteins. Genetic engineering, on the other hand, describes the process of manipulating genes. It can be outside of a cell's or organism's normal reproductive process. It generally involves the isolation, manipulation, and subsequent reproduction of stretches of DNA into cells and is usually undertaken in order to confer on the recipient cell the ability to produce a specific protein, such as biopharmaceuticals. That is, when a gene is inserted into a cell of an organism, the gene is expressed to a protein molecule. In case of recombinant DNA technology, is a piece of DNA artificially created in vitro, which contains the DNA, either natural or synthetic, obtained from two or more sources. So the recombinant DNA technology has been widely used in a range of areas, such as production of therapeutic products or production of genetically modified products. It is also used for the diagnosis of diseases and also in the production of biofuels. So in case of the vaccines, which is widely used in today's world, the COVID vaccine, which introduced by the Pfizer, is a recombinant DNA product, as we know that this is a messenger RNA vaccine. Also, in case of diagnosis, so recombinant DNA technology has been used to diagnose the human genetic disease. Also, it is noteworthy to mention that the diagnosis or the detection of COVID infection is also being done using a recombinant DNA technology such as RT-PCR.